So what we did in this village was really, we cheated in that we just went into huts where we knew there was at least one person that was infected. And then what we did was collect resting mosquitoes in the morning, okay? So they had fed on the people in the complexes, and then we collected these mosquitoes and brought them back into the lab. And the gentleman on the right is Henry DeGoro, and I'm sorry to announce he died last summer. He was just this really cool guy that we worked with. So that was sad. But we do the dissections then, and this is not elegant biology. This is looking at what the parasites do in these mosquitoes. And what we found was pretty exciting for a couple of people that had spent their life working with melanotic encapsulation, is that these things melanize parasites really, really well. And they do it quite well. In fact, we dissected 418. Here's one of my data slides. And we got 72 infected, which is pretty good for an area that had lymphatic filariasis eliminated. And we recovered 242 worms, and about 50% of them, of the mosquitoes, harbored melanized worms. And so really we killed about they were killing about 22% of the entire parasite population by melanotic encapsulation, and 14 of the mosquitoes were totally refractory. They had melanized everything. So this raised a whole bunch of really intriguing questions that we had. One is that obviously all of the work they had done before with PCR diagnostics in mosquitoes, you couldn't really use any of them as they had in the past to determine what was transmitting, because many of these mosquitoes undoubtedly had parasites that they had killed, and they were just detecting residual DNA from these dead mosquitoes. The other thing we had is this is one mosquito, and we don't know about any of the others. What do they do? In fact, nobody in Papua New Guinea in all these years has actually done controlled feeding experiments with mosquitoes to see what percentage of a population is even susceptible to infection. The thing that really intrigued us is this third point, because these mosquitoes also ingest malaria parasites. How would this, if they would ingest both at one time, the filarial parasites go through the midgut right away, and this reaction starts almost immediately. And then about 24 hours later or 30 hours later, the malaria parasite comes through, and the question is, if we activate this immune system, is going to whack the malaria parasites. Because a number of people have suggested in other areas where Anopheles transmits both parasites, that when you have both parasites, you don't have nearly a problem with malaria as you do if you don't have Wuchereria bancrofti. Other people dispute that, but still it's kind of neat. So this kind of intrigued us, this thing. And if you look at the background on malaria in Papua New Guinea, there's lots of it. There's very high transmission in most of the areas along the coast, and seasonal transmission in the highlands. But virtually everybody in the villages that you talk to have had malaria at one time. If you look at a very recent article where you actually look in humans using PCR diagnostics, this is some stuff that came out of Pete Zimmerman's group at Case Western Reserve University. This is pretty amazing. It's like very few people only have a single parasite species as an infection. And you have, almost 30% of the people have Wuchereria bancrofti, and at least one species of Plasmodium, and oftentimes two species. And you say, well, why didn't they know that before? It's because nobody ever talks to each other. And besides, if you were going out and bleeding somebody to determine if they have malaria and doing a blood smear, you're sure not going to do that at midnight to see if they also have microfilariae. No self-respecting malariologist is going to go out at midnight and collect samples. He's back in the cabana having a beer. He isn't going to do that. So, and likewise, the people that are doing filariasis work sure aren't going to be checking their individuals for malaria at the same time, because it's a very different thing. You were spinning the blood down to try and find the parasites. And if you go into some of these areas where you're doing microscopy on thousands of slides of malaria-infected people, if you read a slide and you find banana-shaped gametocytes and you say, Plasmodium falciparum, you don't keep looking at that slide and say, now let's see if we can find another species of Plasmodium. That doesn't happen. So if you think about it, these results are probably surprising from the standpoint that we didn't figure out sooner and anticipate that this type of thing might be happening. So this took us back to this system again that we had, because we wanted to test this in the lab, because we weren't going to be able to test it in the field until we got all kinds of permission to use humans as subjects, because these parasites don't go into anything else, okay? They're only parasites of humans, so you can't use a rodent model. But with Armigeries and Brugia, you can, because you've got gerbils, Mariones unguiculatus, that function as a very good host for this human parasite. And Armigeres is a natural vector for Brugia pahongi, and it melanizes Malaria just like what was happening in Papua New Guinea. And Armigeres is also a natural vector of an avian malaria in Southeast Asia called Plasmodium gallinaceum that infects chickens. So we use this. And one of my doctoral students, Matt Aliotta. So here you have these cute little chicks and you have cute little gerbils, and they actually harbor these parasites. We treat them very nice, okay? Well, not the chickens, because they get the parasite infection and it's fatal, so we have to euthanize them. But gerbils live out their life in our lab and die of old age. So you can do all kinds of different manipulations. You can feed on one and then on another and do the appropriate controls. Or what we wound up actually doing was collecting microfilariae, concentrating them, and then putting them into the blood from a chicken that had Plasmodium gallinaceum and exposing mosquitoes to both parasites at the same time, and then had our controls to see whether or not they had any effect. And we were real excited because we wanted our innate immunity of the mosquito to kill these things, okay? And so the first experiment we did, we went whoopee, because in their melanizing these Brugia malayi, we had a reduced, I'm not going to go through these. The sum story is that you have a significant reduction in both intensity and prevalence of malaria parasites when they take a blood meal that also contains Brugia parasites that are killed by melanotic encapsulation. So we got really excited, Matt got really excited. So then I said, well, you better do this with a parasite that doesn't get melanized, Brugia pahongi, and he did that, and the exact same thing happened. So we also reduced the intensity. So I don't, as much as I want to, it has nothing to do, at least with the immune response of melanization. Now, it could be due to the fact that the entrance of these worms into the body cavity stimulates all kinds of responses that maybe don't kill the filarial worm parasite, but may have a negative impact on Wuchereria bancrofti. So this then, raises these questions. Does this apply, to endemic areas for the real parasites? So can we take what we did in the laboratory and take it to the field? And if this does occur in the field, then we have to sit there and think. Then you have a situation where you could indeed enhance transmission of a much more dangerous parasite in terms of killing people in Plasmodium falciparum than what you have in Wuchereria bancrofti. So we're trying to find this out now, thanks very much to the NIH and Fogarty that gave us money so that we can actually do this project. So Sarah Erickson is in Papua New Guinea now trying to do this, and they're not trivial experiments because none of these mosquitoes are colonized. So you have to try and either develop colonies or else work with lab-collected larvae or field-collected larvae you bring into the laboratory, and then you have to time all of this with being able to go into the field, spend the night in a village, get your night bleeds from somebody that has a microfilaremia, bring that into the lab, and hope that you get somebody coming into the clinic next door with malaria at the same time so you can do your mixed infections. So it's a lot easier to do it in the laboratory. But that's what makes field studies so much fun. You can always have an excuse for not getting something done, as you well know, okay? Regardless of whether this is or is not, I think it points to a really critical consideration, and it gives arguments for integrated disease control that we as scientists are absolutely horrible at. Schistosome people do schistosome, trachoma people do trachoma, HIV, AIDS people do HIV, AIDS, malaria people, and never do they talk to each other about how they might all get their tools together in one village and do something that's a little more holistic in what they do. So this has been argued by a lot, okay, that we do an integrated, neglected tropical disease control, and it's probably being argued by people like Peter Hotez about as much as anything else. But it's easier said than done because we all have our own agendas and we all have our own timetables and we all have our own funding, and it doesn't necessarily overlap with other people. But I think it is a much more cost-effective pro-pore package of preventive chemotherapy that could be done. And I think most of us realize that. And if we can tie in with the big three, which is malaria, tuberculosis or HIV/AIDS, it makes it much easier because they're the folks that have the majority of the bucks. Okay, they can have a nice truck to get you there and it won't break down. Okay, so that's nice. Okay. But to go into this, in Papua New Guinea, they have the Pacific elimination of lymphatic filariasis, and they also have a global program to eliminate malaria. They got $120 million, so this is a BedNet study, okay? And so actually on the Solomon Islands, they actually eliminated lymphatic filariasis accidentally because they were trying to control malaria, and in so doing, they eliminated lymphatic filariasis. So there's lots of neat things. But there's some things you have to consider when, bed nights are like these gold standards. So John Bosco Kevin and some others did some really neat stuff. They did before bed nets and after bed nets, they did landing catches. So they went out and they did a lot of mosquitoes. 35,000 anopheles is a lot of mosquitoes to collect. So they did the identification, did the confirmation. So what they were doing is actually doing time collections to determine when did mosquitoes actually come to be collected. So if you look at Anopheles fawadi or Anopheles punctulatus, and this is in 1 year. And so the blue line is what it was like before they used bed nets, as far as when the mosquitoes fed at what time of day. And the green, I'm colorblind, it's like the greenish or yellowish line, okay, is a year after using bed nets. And what do you see? You see this shift in the behavior of the mosquitoes so that they're feeding earlier. So how good are bed nets if they're feeding before people go to bed, okay? And this has been a question that a lot of us have had, and it just tells you that one thing isn't going to necessarily work, and getting the heads together of a lot of different people is probably a pretty good idea. So I don't know where this is going to go, but we're having fun going, and these are a bunch of people that have helped, and these people in particular have been very helpful in the stuff that I presented. Now, I'll answer questions if you have them.